I'm Nidhi Kumar and you're watching Science Time, a show that brings to you the best that science offers, from exciting developments in science and technology to futuristic solutions. Quantum weirdness could be the driving force behind DNA mutations. Magnets made by soil bacteria offer hope for breast and prostate cancer. JNU biologists identify a new way to better treat Kala Azar. And let's take you to story number 1. Genetic mutations are changes in the DNA of an organism that can result from errors made during cell divisions, viral infections, or exposure to radiation or carcinogens. They are essential to evolution as they can lead to adaptations that allow certain organisms to outcompete others in their environment, but can also lead to disease. They can be thought of as spelling mistakes in an organism's genetic code, DNA. Is made up of 4 bases A, C, T and G that under normal conditions always bond together in specific ways. These bonds form the rungs of the twisted ladder that makes up DNA's iconic double helix structure. However, if the nature of these bonds becomes altered in some way, the normal pairing rules break down, leading to incorrect bases becoming attached to one another and possibly giving rise to a mutation. Now, researchers from Surrey University have found that this mismatched bonding may be caused by a mysterious phenomenon known as quantum tunneling. Tunneling occurs when particles move through a barrier that, according to classical physics, they shouldn't be able to via quantum effects. In the case of genetic mutations, the Surrey team found that proton subatomic particles involved in the bonding of DNA are continuously tunneling back and forth across the energy barrier found between the two sides of the helix. If they do this in the moments before the helix splits along its center during the first stage of the DNA copying process, some of the protons can get caught on the wrong side. This can lead to an error in copying and potentially a mutation. And with that, let's look at story number 2. Scientists are developing magnetically guided microscopic projectiles that can be injected into patients' blood to attack breast, prostate and other tumors. The project, led by researchers at Sheffield University, builds on progress in 2 key medical fields. The first involves viruses that specifically attack tumors, and the second focuses on soil bacteria that manufacture magnets which they use to align themselves in the Earth's magnetic field. The anti-cancer viruses that are being exploited by the Sheffield researchers are known as oncolytic viruses. They occur naturally but can also be modified to improve their efficacy and to limit the chances of them infecting healthy cells. After infection with an oncolytic virus, a cancer cell will burst open and die. The US Food and Drug Administration has already approved the use of TVEC, a modified herpes simplex virus that infects and kills tumor cells and is now being used to treat people with certain types of melanoma, a skin cancer. However, the Sheffield team wants to expand the range of tumors that can be tackled this way. In particular, they want to target breast and prostate cancer as priorities. Some species of soil bacteria synthesize iron oxide nanoparticles that are called magnetosomes. These are used as compasses that allow the microbes to navigate in the Earth's magnetic field and help them find optimum conditions for their growth and survival. And with that, let's take a look at story number 3. Kala Azar is one of the biggest health problems identified globally, which, according to the World Health Organization, is fatal if left untreated in over 95% of cases. It is characterized by irregular bouts of fever, weight loss, enlargement of the spleen and liver, and anemia. It remains one of the top parasitic diseases with outbreak and mortality potential. Leishmaniasis is caused by protozoan parasites which are transmitted by the bite of infected female phlebotomine sandflies. According to WHO figures, an estimated 7 lakhs to 1 million new cases occur annually. The study, led by Professor Anand Ranganathan of the Special Center for Molecular Medicine at JNU, along with a team under Shelja Singh of SCMM, exploits a green chemistry-based approach to chemically synthesize glycoside derivatives. These are composed of a glucose entity to enhance efficacy against the strain of Leishmania donovan. The study has been published in the Journal of Frontiers in Cellular and Infection Microbiology. Kala Azar is a dreaded disease and endemic to eastern parts of India. If this new molecule can in any way combat this disease, it could prove to be a better and cheaper alternative to the currently used amphotericin B, said Professor Anand Ranganathan of the Special Center for Molecular Medicine, JNU. And with this, we come to an end of this edition of Science Time. We'll be back with more exciting stories from the world of science next week. Until then, stay tuned to India Science. Keep watching India Science. Namaskar.